Hi, welcome to the Bloomsbury SET conference on infectious disease and antimicrobial resistance. So this is session 5. The title of this session is Applying Innovation and Knowledge Exchange Approaches to the Field of Emerging Infectious Disease. So it reminds me that emerging infectious disease reminds me of COVID-19. So this session is highly relevant to our situation today. So the Bloomsbury SET conference has been going on from Monday, and it finishes tomorrow with the last session, session 6. It's about the future of Bloomsbury SET. So in this session, we'll have a panelist and talk about innovation and knowledge exchange approaches. So I'm Dr. Ying Chen. I'm the head of research and knowledge exchange at SOAS, University of London. So SOAS is a specialist arts and humanities social sciences college in the Bloomsbury SET. So we are part of this set for infectious disease and antimicrobial resistance. This is because we're trying to promote interdisciplinary work in this area. So joining me today in the panel, we have Dr. Martin Smith. He's head of Manufacturing Science and Technology, Vaccines Manufacturing and Innovation Center. And we have Dr. Sarah Cordery. She's the grant funding officer of GamReef. And we also have Professor Paul Davis. He's the chief scientific officer of Malogic. So for the audience, this session is going to be recorded and will be available on the Bloomsbury SET website post the event. And please do sign up for our closing session tomorrow at 11. And for this session, so we'll have presentations first and followed by panel discussion. So please post your questions in your chat box as we progress with the presentations. And we will pick up your questions in the chat. And so the panel will be answering your questions or talk around your questions. So without further ado, I'm going to introduce Dr. Martin Smith for his presentation. Martin?
Thank you, Ying. Thank you very much for that introduction. Well, it's a real privilege here to be able to speak to you today about the Vaccine Manufacturing and Innovation Center, which I'll refer to throughout this next 10 minutes or so as VMIC. And it seems like there's never been a more fitting time to talk about vaccines. And although I've entered into this field for the second time in my career, I am looking forward to a time when I don't have to talk about vaccines. Having said that, let's talk a little bit about what VMIC is and how it came into being. So if we could move on, please, to the next slide. There we go. Excellent. So what is VMIC? So VMIC has been in sort of conception since 2015, 2016, when it was recognized that the UK had a somewhat uncoordinated approach to vaccine development and manufacturing. And there was a certain awareness, even back then, that if we were to be faced with an epidemic or a pandemic, then our response as a country would be somewhat lackluster. So a number of academics spread between Oxford University, Imperial College, and the London School of Tropical Hygiene and Medicine came together to form a partnership to start planning for a relatively modest, small-scale facility and organization that could help accelerate the development of vaccines from the bench to the clinic. And as that partnership gathered some pace and plans began to be developed, a number of partnerships were formed with industry bodies, such as Cytiva, formerly known as GE Healthcare, Merck, and Janssen, who came on board with this partnership and started to expand the scope and the mission for VMIC. Around 2018, we received funding from Innovate UK to establish VMIC as part of the UK national scientific infrastructure, and that's when planning really started to take shape. And VMIC really will be bringing to the UK some quite state-of-the-art capability in the development of vaccines, and it will be, as I'll show you in a subsequent slide in a few minutes, quite a large GMP facility, a facility that is compliant with the latest legislation and regulations for producing vaccines. The goal is to really kickstart, once again, the vaccine industry in the UK, and to attract vaccine developers across the globe into the country, where we can start to further develop a highly skilled workforce in this particular sector. And also of great relevance at the moment is for VMIC to be able to provide a rapid and immediate response to emerging threats, be they epidemics, be they pandemics, or be they any other emergencies that require vaccines to be manufactured. And more than any of those, we're also going to try and create a great facility that is an inspiring place to work. Could we move on to the next slide? The facility itself is being built just outside of Oxford on the Harwell Science campus, and in fact next to a decommissioned nuclear reactor, which can sometimes be a little bit eye-raising. It's on a campus of around 700 acres with a huge number of companies, including the Diamond Light Source, which we've already started to have early discussions with about potential collaborations. If we move on. So this investment by initially the UK government is to create a not-for-profit organisation, which we hope will be recognised globally as a centre of excellence for vaccine development. And we're certainly aiming to be a key opinion leader in vaccine manufacturing and development. I'll move on to talk a little bit more in later slides about our emergency response capability, as it has changed from where we originally had positioned it in 2018, 2019 to where we are now. So I'll cover that in a later slide. So if we could move on. So VMIC has some long-term strategic goals. We're a new organization with a new team, and we recognize that it'll take us some time to achieve our mission and our objectives, but we really want to create an environment where we can ourselves and in partnership with academics and industry partners excel in research and development and in innovation, and to do so through long-term partnerships where possible. We are establishing vaccine process and analytical development to move vaccine candidates into early phase clinical trials as quickly as possible. And this last year has really shown everybody involved in the vaccine space and indeed biologics development just how quickly we can move when we have the platforms in place and the need to do so. The facility itself will be licensed, hopefully, well, certainly it will be licensed by the MHRA towards the end of this year, and that will allow us to continue to move those products very quickly through into clinical trials. We've already talked about emergency response, and I'll come back to that in a few minutes, and also talk a little bit about how constructing this facility and building the team will allow us to train the next generation of life science graduates to enable them to either stay with us at VMIC and help us translate vaccines through the facility, or potentially move on with those vaccines once they've been in our facility to other manufacturing sites that will hopefully be established throughout the UK. On the next slide, I'll show you a little bit about the facility itself. Now, you may notice that the image is slightly degraded in quality, and that's somewhat intentional. The facility itself is around 75,000 square foot, or around 7,500 square meters. And it's roughly broken up into 5 areas. Towards the right-hand side of the floor plan that you can see on the screen in front of you, there's a sort of white rectangular area. That's our entrance and office and administration areas where the staff will sit. And there are some hospitality areas there. Towards the left-hand side of the building, there's another white zone. Those are some utilities and also quite a large warehouse for all of the raw materials that are needed to make vaccines at various scales. And there's also some finished goods storage in that particular area as well. But at the bottom of the layout, you can see an area highlighted in yellow. That area highlighted in yellow is a series of relatively small 40 or 50 meters squared clean rooms, which can be used independently and in parallel to make up to 6 different types of vaccines at the same time. These rooms would typically be used for a very fast translation of laboratory scale products into phase 1 clinical trials, where the overall number of doses required is relatively small, and they can be made in relatively small pieces of equipment and therefore relatively small rooms. The green zone just above that is a series of, I think in total there are 8 clean rooms of various levels of cleanliness. Our most clean rooms are called grade B, and we have 2 of those clean rooms, and there are 6 grade B clean rooms, grade C clean rooms. We will be installing into these rooms initially some suspension bioreactors for growing cells in vats of around 200 and up to multiple 1,000 litre bioreactors. And those types of bioreactors and those scales of operation are currently being used to address the current pandemic. So when we come on stream later this year, we'll be able to either contribute to the current pandemic if it is still ongoing. It certainly will be in certain areas of the world, unfortunately. And we will also have put in place by then a significant capacity housed in the UK for rapidly responding to the next pandemic. Now, the area in green and yellow produce what we call drug substance, which is typically a liquid product in a bag that still needs to be converted into a vialed product that can then be distributed throughout the country and the world for administration. So the area in blue is our fill finish portion of the facility, where we have a number of high-speed filling lines that can take that bulk drug substance and turn it into literally hundreds of thousands of vials in a matter of hours or potentially days. I'm not showing you on this slide the first floor, where we also have some process and analytical development laboratories and our QC labs, and also all of the mechanical side of running such a facility, such as the HVAC, the power, the steam, the water, and so on and so forth. But I hope that gives you a sense of what we're intending to build, what is currently under construction, a very large facility that could potentially run up to 14 different vaccines at once if we have the demand, or could consolidate all of its resources and focus on pandemic response. And we're certainly building the flexibility to do both. If we could move on, I'll tell you a little bit about our role in the current pandemic. I mentioned earlier that planning for VMIC started in 2018, and at that size, the facility was funded to the tune of around 65, 66 million pounds, and was significantly smaller than the floor plan I've just shown you. But of course, in February, March next year, we were approached by the UK government, and a sensible investment decision was made to increase the capacity of the facility up to 20-fold. So the original plan was to produce roughly around 2 to 3 million doses in the event of an epidemic in about 4 to 6 months. We've increased that capacity now to be able to produce up to 70-ish million doses in around 4 to 6 months. And that will vary a little bit depending upon the technology that's used and how productive this is. Not only have we increased the capacity, but we also rapidly decreased the time of construction of the facility. It was due to come online at the end of 2022, and we're now aspiring and planning to bring it online at the end of this year. And that's resulted in a need for significant increased funding, which was provided by the UK government earlier this year, the middle of last year. We have been involved in the current pandemic. We've helped establish a rapid deployment facility, which has been referred to as virtual VMIC, but it also is more appropriately referenced as the provision of equipment to Oxford Biomedica, who we've helped enable come on stream far more quickly than would otherwise have been possible. And as you'll have heard through the press, they are now currently partnering with AstraZeneca to make the vaccine in their facility. So we've helped provide some funding from our initial investment and some trainee staff to help them meet these goals. If we could move on to the next slide. Conscious of the theme of this particular conference today, I thought it would be a useful slide to put in here to help us reflect on what's really changed over the last year and what's been enabled by at least 10 to 20 years worth of basic research and applied vaccine development. And that's the difference between the traditional approach for bringing a biologics to market, and certainly it's true for a vaccine, versus where we found ourselves in the last 12 months. So at the top of this slide in the box that I've taken from a New England Journal of Medicine reference, which is article there, you can see the very measured stepwise approach for bringing a biologics or vaccine to market, which starts with target development, early exploratory studies at the small scale at the bench, where a production process is designed at a very small scale. And once that knowledge has been acquired, it's then scaled up into larger equipment, and small amounts of product are made for phase 1 clinical trials. As we've all become armchair experts in this over the last 12 months, you'll know that that's to test initial safety. We once again then take that knowledge, assess it, review it, scale up again, expand the number of doses, test it in a wider number of people. And then as those results come through and companies look at the commercial risk that they're exposed to, they scale up again. And further knowledge is acquired and consolidated, and phase 3 trials are completed, and then products are submitted for late-stage licensure and then commercial manufacturing. All of that changed in this current pandemic. Now, decades of research by companies such as Pfizer and institutions such as Oxford University have put in place the framework for rapid response to pandemics by designing rapid vaccine platforms. And advances in genomics have allowed us to be able to sequence a virus in literally hours and then incorporate that into a vaccine platform also in literally hours and days, and to immediately start bench scale studies and transition very, very quickly into small scale manufacturing. But that means moving ahead without all of the knowledge that you would traditionally have acquired and sequentially applied to reduce commercial risk. And it's primarily around commercial risk. It's not at all about safety risk. So that's what producers of these vaccines have had to battle with over the last 12 months, is moving ahead without having the complete picture of all of the knowledge that we would typically acquire over 10 years, and moving ahead in literally less than one. And by risk assessing those knowledge gaps and working out how and when it's appropriate to acquire them. And the other thing that's happened, of course, that's of relevance to VMIC and VMIC's construction is that to rapidly produce 70 million doses has required outsourcing of process development, outsourcing of manufacturing, and outsourcing of vialing of these products. And for some of the big pharma players, that's involved doing so at multiple production sites around the world. What VMIC will bring to the UK is a centralized facility and organization where all of that can be done in one place. And that has huge advantages in terms of the centralization of knowledge and leveraging the speed with which you can move knowledge around an organization without having to translate it into different countries, and actually using the word translation, translating it into different languages as well. All of those things will still be possible. I think all of those things will still come to bear in any future pandemic, but we're certainly building in the UK a really useful arsenal to help us address this, either next year if needed, or in future in the next pandemic in a more efficient way. If we can move on to the next slide, I'll show you a picture of the facility. This was taken approximately a month ago, I think. And you can show that construction from the outside at least is well advanced. This facility will still take the rest of this year to construct and install all of the equipment that's needed and qualify it, and then have it licensed by the MHRA. But it's well on its way, and it's a fantastic facility. I'd encourage you, if you have the time after this conference, to move on to the next slide, where we have a link to the website. And on the website is a really quite insightful time lapse of the construction of the facility. It's well worth watching, and you'll get a sense for just how big it is and just how quickly it's been constructed. And do please feel free to get in touch at any of the links above if you wish to talk to us about partnerships, or if you just generally like to hear more information about VMIC and its mission. And with that, hopefully I've kept reasonably to time. I'll draw to a close. Thank you.
Hello. Thank you very much, Martin, for your wonderful talk. I think we all feel reassured that we have such a large facility that's going to be ready so soon to manufacture more vaccines for the pandemic. So our next talk is from Sarah. She's talking from a founder's point of view about funding, innovation, and knowledge exchange. Sarah, over to you.
Thank you, Ying. So GEMRIF is part of Global Health Security, which is within the Department of Health and Social Care. We fund early stage R&D, so specifically to reduce the threat of antimicrobial resistance. We are a relatively small fund with 50 million allocated when GAMREF was created in 2016, though we've leveraged significant funds on top of that since then. Next slide. So GAMREF only spends UK aid money, also known as official development assistance. Therefore, projects must directly and primarily benefit people in low and middle income countries. So obviously that would determine which diseases or indications we're interested in targeting, and the maximum cost of goods or the maximum anticipated cost of goods. But other considerations such as cold chain, or which route of administration is the product going to be. Obviously, a tablet is better than an injection or an infusion, so on. So I need to credit IDRC for this photo. Photos just showing culture and sensitivity testing of a patient sample. IDRC is the Canadian International Development and Research Centre, and that gives me a chance to plug their really excellent series of podcasts on AMR for those people who want to dig a bit deeper into AMR. Just Google IDRC podcasts AMR, it will come up. Next slide. So this graphic shows the intervention types that GAMREF actually funds. So the emphasis here is by the number of projects as opposed to the amount of money for each intervention type. So you can see that we fund tangible products such as vaccines and diagnostics and bacteriophages. In case you're not familiar with that term, that just means viruses that only attack bacteria. And we also fund non-tangible research, such as research into optimal agricultural practices with the goal of reducing antibiotic use. And we also fund research into cloud-based data collection to capture information from point of care diagnostics. So that's useful for surveillance and disease outbreaks, but also countries can make up-to-date data-driven decisions on where to spend their healthcare budget. Another strategy to tackle AMR is just to make more antibiotics. So that's fine, but just a couple of points related to that, which people may have heard this a million times before. But to be clear, that there's no point in making new antibiotics without a stewardship plan. And by stewardship plan, I mean, for example, making sure that there is a cheap and easy to use diagnostic for the disease that your antibiotic treats. Otherwise, the antibiotic will be overused and you're just going to be in the same position 5 years later, and bacteria or whichever pathogen is going to develop resistance. So the second point about why not just make a new antibiotic is that this is an incredibly expensive endeavor. Unfortunately, I don't have a value for veterinary antibiotic. I only have for human use, but recent estimate is $1.7 billion all in. So as we know, antibiotic resistance, antimicrobial resistance, antibiotic resistance, driven by antibiotic use, we're asking the pharmaceutical company to invest their 1.7 billion, and then we're going to say, but we don't want anyone to actually use this antibiotic unless they absolutely have to. If you are interested in the market forces and the regulatory angle of how to tackle AMR, John Rex does a really excellent newsletter about this. Again, just Google John Rex AMR. Next slide. So who actually does all the work for us? We have 7 delivery partners arranged under different types of partnership, which this slide is trying to capture. The research is carried out in academia as well as in the private sector. On Monday, Emma explained how Bloomsbury Set takes a One Health approach. So just to reiterate, in the context of AMR, that's generally taken to mean that you can't fix the problem of AMR without addressing the 3 different areas of human health, the environment and animal health, particularly food production, so land-based as well as aquaculture. The latter is rapidly growing area, particularly in Southeast Asia. And the Gamma portfolio, as I hope shown here, does cover those 3 One Health approaches. Next slide. So to bring it back to the session title in the last couple of minutes, as well as giving our researchers cash, most of our partners give wraparound support to the researchers, and that's built into the whole package. So for example, in our Innovet program, the researchers receive tailored training from consultants actually called Wellspring Development. They specialize in agri-science, and they provide, as I say, tailored training in commercial skills. For example, an academic may not be familiar with the steps involved in taking a product all the way to market. And the researchers also receive training in intellectual property, and there is a dedicated social science consultant available as well. Our UK-Argentina partnership, which is managed for us by BBSRC, comprises 5 different projects, and each of which has 2 components, the UK researcher and the Argentinian researcher. So after the funding was awarded, we asked these 10 different groups of researchers to get together, and in this context, I mean get together physically in the same space, to draw up a plan with how they would like the 5 different work streams to be synergistic, and more specifically, how they would make sure that their outcomes, which will be policy recommendations regarding agricultural practice, how those outcomes will be not only applicable to Argentina, but also wider than that, let's say Brazil and Colombia, and outside Latin America, Kenya, India, Bangladesh, other countries which have a significant agricultural component to their economy. So the overall consequence of this plan, which the researchers themselves put together, is that we get greater value for money from our funding in terms of impact. Lastly, on this slide, I'll just point out Back2Vac. So that's a bacterial vaccinology network. And in this case, the organization provides help to the researchers at the pre-award stage. So, for example, you may be trying to fill in an application process when English is not your first language, and you don't want to miss a great research project for the wrong reasons. Back2Vac also support their researchers with skills such as contract writing. So the idea is that as well as investing in the R&D, Back2Vac are also investing in the researcher and in their institution. Next slide. So I'll just leave you with this image from a project that we fund in Malaysia, where the aim is to reduce the incidence of bacterial infections in commercial shrimp farming. Thank you for your attention, and I will hand back to Ying.
Thank you, Sarah. for the nice introduction of GAMRIF. It's so good to see that Department of Health and Social Care has a foresight and investing in this area. So next we move on to Paul. He's going to tell us the Mologic story. Paul, over to you. Thank you.
Would you believe I was joking before we started about people leaving their mute button muted, and I was victim or guilty of doing so. I apologize. So now you don't have to lip read. Thank you for the opportunity to tell you about the Mologic story. And it so happens that the title of this session almost perfectly encapsulates what the Mologic mission is. The Mologic mission is all about applying innovation and knowledge exchange approaches in the field of emerging infectious disease. I guess we go a little bit wider at times, but Mologic is deep into the heart of this sort of activity, and I should explain what that means. Very briefly, Mologic was founded in 2003, and it's a father and son enterprise. So my son and I started the company with the grand total workforce of 1.2 people. That was all of him and 20% of me, because I was doing other things at the time. And we started small, and we have grown substantially. We never really thought we would. And the next slide just shows you the home of Mologic at the moment. And in this slide, you can see that Mologic has its name tied up with some buildings that may look impressive in this picture. They're not terribly impressive, but they're on what used to be a Ministry of Defense airfield, where the Harrier jump jet was developed, and it was since abandoned and taken over, and we've been able to take over buildings in that site. And most of those buildings that you see there are now occupied by Mologic and its manufacturing group, the associated manufacturing group of Global Access Diagnostics. I've highlighted a couple of products there which we were developing earlier in our life and are still highly relevant. One of them is to do with personalized self-testing in chronic obstructive pulmonary disease, where we've developed a multi-biomarker test for predicting the onset of exacerbations in COPD. And it allows patients to look after themselves more, because they can track their own disease status by daily testing for these biomarkers. And they don't need to know what the biomarkers are. They just get a warning that they're heading for an exacerbation episode, so they can take early steps to control that exacerbation. And another one, which is called Periplex, which is all about testing patients' urine. Well, actually, it's not testing for urine in this case. The COPD one is urine. The Periplex one is by testing the effluent from dialysis in patients with end-stage kidney disease. And that allows them to spot when they have an infection, which can be disastrous in their peritoneal dialysis. So those are things that we had running. Now the next slide tells you about something that happened in 2016, which is that Mologic, which had grown considerably by this time to about 60 people, was approached by the Bill and Melinda Gates Foundation. And they came to us with the challenge of saying, well, lateral flow test, which is what we do, we're really quite big in lateral flow tests, really is a mature technology. It started off with a big bang in the 1980s with home pregnancy tests, and it became widely used and has a great role, but it was sort of stagnating. There wasn't a lot of new thinking, there wasn't much innovation. And they said, could we take a deep dive into lateral flow and come up with new ways of making it more sensitive, more usable, more reliable, give it a kind of rebirth, a renaissance. And the reason for that is that the Gates Foundation wanted to be able to offer better and better diagnostics at the low cost to people in the low to middle income countries. They felt that these populations were being underserved in only having tests which were not really right up to being first rate. And so we were able to set up within Mologic a group called the CARD. The CARD is the Centre for Advanced Rapid Diagnostics. Its job to reinvent lateral flow, to take it to new heights of sensitivity. And my goodness, we need that now, because we keep hearing about why lateral flow tests aren't really up to the job of providing the kind of surveillance and prediction and disease detection as they should. And of course, there's a big story associated with that and what we're doing in order to create epidemic readiness, in order to push the diagnostics further and faster. The next slide is a kind of pictorial image of part of our context, part of our vision, which is that of pandemic readiness. And the world is covered with diseases that need attention. Immediately COVID-19, but there's still hepatitis C virus, influenza, Ebola, and also various other tropical diseases that really don't get the attention they need. And here we were working away in the CARD to try and accelerate and improve these technologies when COVID struck. And in a way, this was like Bill Gates having the foresight that he had to predict that we were going to be afflicted by a pandemic before long. We thought how incredibly foresighted it was to set up the CARD in order to be able to position a response to the COVID outbreak that would have some impact. And just to confirm that, we had a visitation in March last year of none other than Bojo himself. And we had a wonderful time, and I was able to show how we like to use low cost examples. So here I am explaining to him a tangerine stuffed with map markers, mimicking a model of the coronavirus, the map markers being the spike protein and the peel representing the virus membrane. And he was really amused by that, and perhaps he remembered it. Now, the next slide shows you what we're doing, because you might be thinking, well, why aren't we churning out these diagnostics now when we so urgently need them? And here we have a resonance with Martin's comment about the time taken to get things running. When you need to have the investment, you need time to build things, you need a run-in. And there's an important discussion point here, because what we are creating with another company called Flexitronics, a very small company, a whole new approach to manufacturing lateral flow point of care rapid diagnostics. And we're bringing disruptive changes to the way they're manufactured and to the way they can deliver. And you know, with rapid diagnostics like lateral flows, one of the ways to make them really effective is not just to make them individually more sensitive, but at a scale and a sensitivity and a cost, abundantly clear, a cost that makes it possible to use lots of these tests. It's not very helpful to test a child at school once a week. You need to test that child once a day in order to make that test work. And it will work extremely well, even if it isn't as sensitive as a PCR, if you can repeat the test day after day. We had a small outbreak in Mologic not so long ago, and we had everybody testing twice a day, and that allowed us to control that local epidemic, local outbreak really well, because everybody had access to tests. It didn't cost them anything, and they tested twice a day. And so what we're all about here in this diagram is really changing the rules so that we can use the investment that was made in this to manufacture at an unprecedented scale and at an unprecedented low cost. That's what we believe is an industrial revolution about to happen, full of risk, full of uncertainty, but with funding made available to us. That's what we're doing. Next slide now just focuses, I've been talking about innovation. Now I'm going to talk about transfer of knowledge. And the Mologic vision is to be able to share our knowledge and innovative technology across borders and without profit. And what we're doing there is building local manufacturing facilities, not just to do this extreme manufacturing, but also, when I say local, I don't mean just local to Bedfordshire, I mean local to Africa. So the next slide just shows a particular point, which is that when you look at where diagnostic tests are manufactured, they're often being manufactured in places that don't need them, and they're exported to places that do need them. So we've been privileged to play a significant part in setting up an outfit called the Diatropiques. That means diagnostics in the tropics. It's in the grounds of the wonderful Institut Pasteur in Senegal, in the capital, Dakar. And we've been working very closely with them, increasing access to diagnostics through technology transfer and local production. The next slide shows you the exciting new building. So there's two new buildings that we looked at today. This one is in the campus of the Institut Pasteur in Dakar, and it's a new building that has been funded not so much from industry, but from various funding sources like the European Investment Bank and the Bill and Melinda Gates Foundation. And you can see from these pictures, that I won't take you through in much detail, this new building in an area that would otherwise be considered to be not a particularly strong growth area for this kind of high technology. So the next slide allows me to introduce one of the drivers of this technology who we've been able to engage with, and this is Amadou Soul, who is the head of the Institut Pasteur. And on the right, you see the beautiful Art Deco building of the Institute itself. And it's right next to that, this new facility, which you see on the bottom left, has been built. It's been built and it's already going in for its first, or getting prepared for its first extension. The next slide shows the core team in that building. And I'm really pleased to tell you that that team and a few others in 2019, the summer thereof, in the Mologic complex. And we were able to work with them and show them our manufacturing practices. And we had weeks and weeks with them where we got to know them and where they got to know us. And we're really geared up to be able to transfer our diagnostic test information, the formulations, the manufacturing, so they can make them the same as we do, but in Senegal, in Africa, where they're needed. And the next slide shows the building as it stands at the moment, and it's already getting ready for its next extension. So that's what we're doing. Now, one other really important point to make is that we've had investment within our place in the Bedford Technology Park for doing manufacture. And we've been able to set that up as a not-for-profit enterprise. So instead of having it just as part of Mologic, it's a charity. It's not run by a board of directors, it's run by a governance board, drawn from people in Africa and involved in experience in pandemics and tropical diseases and global health. And it is separated from the need to pay shareholders, and its profits get plowed back into it. And Mologic itself is gearing up to be that kind of company that will allow us to be disconnected from the norms of economic involvement of a company and its need to make profits for shareholders. That's a perfectly worthwhile thing to do for certain types of companies, but we feel that for us, with the kind of investments that we've been receiving and are placing that we can do that better as a community interest company. That's why I refer to the story so far. We're not quite there yet, but we will be before too long. So thank you for your attention, and I hope you enjoyed that little snippet of the Mologic story.
Thank you very much, Paul. It's such a great pleasure to actually meet you as the inventor of Clear Blue pregnancy test. I must admit, I used it before. So, yeah, and now also to see that your company has grown and you have moved on to other chronic diseases and also to emerging infectious diseases. So it's brilliant to see this. So now we're moving on to panel discussion. So can we have all our cameras on and yeah, microphone. So I have been, sitting here, listening to 3 wonderful talks, and we have founder, we have 2 sort of inventor and manufacturer in the infectious disease area. So just keep me sort of thinking that funding is so important to bring science and research into innovation and into manufacturing, into diagnostics. So hopefully, we can reflect a little bit on that. And also we have a few questions from the audience. I think the first question was for Martin, of course, for the first speaker. People are kind of quite interested to see that we have such good facilities from government investment ready there for vaccines. Can you talk about how ready we are for sort of the future, for the next pandemic? Will we be much quicker in responding if we have kind of COVID-23 or something, or some other infectious disease on the way threatening us?
Okay, I'll do my best. First of all, let's hope there isn't a COVID-23, of course. We touched on the unprecedented timelines that have accelerated from 10 to 15 years down to 1. And from what I've seen, from what's been publicly reported, a lot of even within that 12 to 13 month, a lot of the timeline has been holdups in clinical trials and conducting those clinical trials in places where the disease outbreak is ongoing. And that has complicated and slowed down trials in various areas. And I think we're going to come out of this first year and a half of COVID-19 with a huge amount of learning and a huge amount of knowledge about what has worked, what has not worked, both in clinical trials and also in the rapid vaccine platforms that have been used to create these vaccine candidates. And where have those platforms run into scale-up difficulties, raw material difficulties, filing difficulties, and I think we will reflect on that. Whether we will move from, in a period of 13 months, having, I can't remember what the latest count is, but the J&J vaccine was approved today or yesterday. We have 4 or 5 approved vaccines now, certainly for emergency use. Whether we'll see that timeline half, I don't know. But certainly, for a pandemic response timeline, I think we will be at least more robust next time, if not more quicker. And I think the other thing to say is what we're seeing in terms of international responses. A lot of governments around the world now recognizing, as the UK government has done, that it needs to have its own national response. And yet, again, in the last week, we've seen statements where we are starting to acknowledge that vaccine nationalism is not the correct response, and that we also need to be bearing in mind our obligation to the broader community out there. And I think the other thing that we will learn from this, from an epidemiological, and I can't even say that word, an epidemiological perspective, and maybe it will change for each disease, is who to treat, how to treat, and when to treat, and how to use our vaccine capacity appropriately to treat those most at risk globally rather than those most at risk just at home.
Of course. Yeah, I think global response is definitely something, that's Bloomsbury's set ambition, and also I think also like founders' ambition. I think Sarah, you talk about funding globally, right? Even the money is coming from our government, but the funding that you talk about are quite global. Can you give some perspective from founders' point of view, how prepared we have been and or we will be about, from diagnosis to treatment on infectious disease, on emerging infectious disease?
You mean how prepared we are now?
Yeah, or how, yeah.
past
So
year?
what kind of founders are also thinking at this moment for future preparedness as well?
Looking into their crystal balls. Yeah. I think, I guess the idea of platform technologies, that's the reasonably obvious thing to say, but technologies which would be relatively easily adapted to different specific indications, knowing that you won't be able to spot what's coming, but you could have your technologies, your starting point ready in advance. That seems like as I said, sort of a common sense approach to where to put your money without knowing what's on the horizon in 6 months' time or in 5 years' time. Bearing in mind, if you're funding early stage research today and you're working at the normal pace that things develop, it's hard to say what do I need to treat in 5 years' time when my product's going to be ready, or even further in the future than that. Not easy.
Right. So yeah, I know it's not easy. Paul, maybe I can get some of your thoughts about funding and innovation. Like you have a great example, you set up a great example, like bringing sort of innovation into, diagnostic products. And I'm sure funding would have been really helpful or speed up your, innovation. Can you give sort of, another reflection on funding and innovation and bringing to the market?
Yes, that's a very insightful question, if I may say so, because when you look at, for example, our big volume manufacturing that we're currently working on, and that it takes a long time to actually design the machinery and just as it does for vaccines, and to have the production site to the standard that it needs to be. That takes time and it takes a lot of money. We've been fortunate in getting investment to do that, grants to do that. But having got the machine to make these tests at very high volume, and the designs and the knowledge of how to do it, we then face another problem, and that is operating capital. So if, for example, we were to be making 5 million tests per day on a machine, you then got to think about how you can buy enough of the materials that you're going to have to put in that machine. When you turn it on and it starts running, it will be gobbling up relatively expensive materials at a huge rate. We've got to be able to make antibodies on a huge scale to be able to put those antibodies in the tests that will make the test work. And we've got to be able to not only have the actual machinery to do it, but to be able to fund the supply of consumables that are going to make you do it. So that's one of the things we're grappling with at the moment. How on earth can you find 10 million just to buy the nitrocellulose rolls that go to make the strips that make the assays? How on earth can you find enough money to not only buy it, but early on to get the commitment from the supplier to actually make that material available to you. And just as there was the toilet roll crisis when we went into lockdown, the access to things like the materials needed to make the lateral flow test, they're a similar thing. Everybody's, well, not everybody on the street, but all the manufacturers are trying to buy as much as they can. And so we need to be able to compete in that area and guarantee that if we order the material, we'll be able to pay for it. So we not only have to raise money to do the research and to build the building and to put the machinery in, we've got to find the money to start with that will allow us to prime the pump and start making the products that we're going to sell. And that is a big hurdle. It takes a lot of time and a lot of effort to actually secure the cash flow that will allow you to have a smooth, high volume production of tests that are needed.
I think that's probably for vaccines, the rapid kind of manufacturing was also because there was investment, right? There was all the government were putting money into Pfizer or AstraZeneca to manufacture those vaccines, and maybe you can lobby Bojo to give you money for more diagnostic for COVID, and maybe government needs sort of support in the diagnostic area too.
And Paul, one question I have actually for the lateral flow technologies, a little bit similar to some of the vaccine platforms. You change the genetic code and you use the same platform. How adaptable are lateral flow tests? I understand how one might have to buy an injection molding device or other expensive machinery, and the nitrocellulose is perhaps reusable between different antigen tests. How much of your technology is platformable versus specific for each new target? I'm maybe using the wrong terminology, but I hope I'm you're getting the insight to the question.
It's a very clear question. So far, we've found that our antigen tests is able to detect, for example, the Kent mutant very easily. We're currently working on the Brazilian test and the South Africa, not test, the Brazilian virus mutant and the South African virus mutant, and there are going to be others. And for an awful lot of them, we will find that the reactivity of the antibodies will be sufficient to allow us to detect those regardless of the mutations. But we need to constantly watch that. They are adaptable because we can change the antibodies that we're using to do the detection. And as we go forward and learn about where the hotspots are going to be, and I guess we can say that those hotspots in the virus structure will be those that allow it to engage with the cells of the human body. And so it would make sense for us to be sure that we use the sort of bioinformatics to make sure that we identify parts of the spike protein and other proteins of the virus to detect the virus, and that we choose those as being in areas that are not hotspots for mutation, or where the virus wouldn't be selected because of a mutant in those regions.
Okay.
So that's the two things. One, you choose your antibodies carefully to recognize the virus in the areas where it isn't going to do a rapid morphing of its structure. But you are vigilant as well to make sure that you've got other antibodies that you can swap in, which will not actually cause a problem. That, of course, means there will be a slight delay while you get your clinical trials done to make sure that it really does the job. But I think it's probably more of the danger to vaccines than it is to the antibody-based tests.
Yeah. Okay.
We have a question in the chat box, which says, we have seen spectacular advances in the response to an emerging pathogen, but how can we be more effective target surveillance for new pathogens? I don't know whether, Paul or Martin or Sarah like to comment on this one.
Well, in terms of diagnostics, I really do believe that we will be better prepared as a result of this awful experience that we're going through now. And whilst we like to celebrate the way things have been done quickly and interactively, and there are good things you can point out, that within the diagnostics field, the amount of cooperation between companies and between academic groups and companies has been huge, allowing us to move. But I'm very struck by the fact that we're now more than a year in, and the quality of antigen tests that we hear so much about at the moment, the rapid tests, is only now becoming at a level of performance that we really need. And I remember Matt Hancock talking about lateral flow tests way back in March last year and saying these were coming. And they came and they didn't do anything, and they've been delayed. And it's taken us a long time, really, by the standards that we need, to get to the point where the tests are now coming through in a form and at a quantity that makes them useful. And frankly, we're still not there yet, because, as I was saying earlier, to make lateral flow tests work really well, to do the job that they need to do, which is to spot infections quickly, that they'll never be as sensitive as PCR. But if you test once a week or twice a week with them, it's not enough. So we've been late in getting the volume we need and late in getting the sensitivity we need. So going back to the question, what can we do better next time? Well, the very high volume manufacturing systems that we're developing will solve the problems, if we're successful, of making enough of the tests available to use them with a frequency that makes sense. The use case is what it's all about. The use case of testing, but testing frequently. So we expect to put in place these types of machines in Senegal and Pakistan and various centers around the world, so that when the next one comes, we will have high volume manufacturing all over the place. We won't be struggling to bring manufacturing up to what it needs to be. It will already be there. And I think we will be sharper and quicker at getting the quality of antibodies that we need to put into those tests to make them sensitive enough to do the job. And we'll know much more about how to deploy them to make them work, and not have this wrangle about whether they're sensitive enough, because we'll know how to use them.
Good. Sarah, do you want?
Yeah. So in terms of the infrastructure that's sort of sprung up in the last year, let's say around diagnostics, I think that it would be important to try to make sure that that doesn't lapse, if you like, let's say over the next 3 years, as the need for COVID testing will drop away, not to zero, but the numbers will reduce. You've got that infrastructure. You don't want to waste it, if you like. So how to turn that onto other diseases. Obviously, I'm thinking about AMR, and talk about what's going to be the next pandemic. As I said in answer to the last question, we don't know what the next pandemic will be, but what we do know is that antimicrobial resistance is going to cause a problem in our lives the next 10 years and so on. So yeah, rather than all of that infrastructure that has developed very quickly in order to meet COVID, just sort of falling away, to try and keep those systems and co-opt them for other emerging infections.
So there's another question in the chat box for Martin about shipping your vaccines to low middle income countries. I think the idea is to say, is there a scope to develop packaging facilities capacities in the low middle income countries in the model similar to Paul's Senegal facility? Something like, I don't know whether you would like to comment.
I can try. I don't think I'll answer it satisfactorily. At VMIC, we're not planning to ship bagged vaccine at the moment outside of the facilities. We're putting inside the facility itself a vialing machine. So we would ship vialed drug product. Depending upon the vaccine type, there are certain limitations with shipping the drug substance, because it can be the case that it's not stabilized, and that's one of the reasons why some of the latest mRNA vaccines need to be used within 6 hours, is because they're deliberately not, as far as I'm aware, complemented with preservatives. So there's that limitation. But is it feasible to ship a bagged drug substance or even a formulated drug product to another site in the world where it could then be filled into vials or potentially into multi-dose administration devices? Absolutely. And I've no doubt that that's being looked at. And I know that CEPI have projects in mind for multiple dose application systems that are not just vials. But I'm certainly not an expert in this field. I don't know if any of the other panelists have any knowledge of this.
Good. Right, we have another question coming in. It's quite a general question. It says, in terms of innovation, what are the most critical changes in processes needed for the future? I suppose for this panel, we can think about in funding, because also there's an earlier question is asking whether GAMRIF is open to other initiatives, and also whether the funding have been fully allocated at present. So we can also think about public and private kind of funding initiatives, and probably the drug regulatory and approval processes. I mean, we've already talked some of that, but just kind of any more thoughts about what are the most critical changes in the process needed for the future? Sarah, would you like to comment on?
I'll start with the question about GAMRIF. So regarding funding from GAMRIF, the spending review results have not been finalized or made public, so I can't say that much on that, but what I can say is that that's been publicly announced, that UK aid funding is tied to this legal target of 0.7% of gross national income under normal times. That's just recently been reduced down to 0.5% due to extraordinary circumstances, plus the fact that gross national income itself will have plunged. However, regarding funding, I don't know if everyone listening to this session will have attended Wednesday morning's session. I think her name is Jo Frost from Innovate UK. She gave a really nice outline of EU funding, which is available to UK researchers, as the UK is counted as an associate country for those purposes. So that's not specifically antimicrobial research funding money, that's science and technology money. So just a plug to go back and watch Wednesday afternoon session if you didn't already see it. But regarding innovation, critical changes needed for the future, well, depending on what you're meaning by innovation. So let's assume you're not talking about lab bench workshop type innovation, talking about broader approaches. Just to mention call it an intervention in the sense of where market forces are not having the desired effect, let's say. I'm not sure if people will have heard of this initiative with NICE and the NHS. Some people call it the Netflix subscription model. So in short, as I mentioned earlier, companies are not investing huge amount of money in antibiotics because they're not going to get those volume of sales, because we don't want to use lots of antibiotics. That's fine. So the idea of this innovation in terms of how you incentivize the pharmaceutical companies to spend their money on this is that the payer, and I know this is unique to the UK way we do things, the payer being the NHS, would pay the money to the pharmaceutical company for an antibiotic that they might never use. It's a subscription, hence the sort of nickname or explanation of the Netflix model. So I'll just sort of throw in a similar analogy to the Netflix model. You may have heard people talking about the fire extinguisher analogy of antibiotics, meaning it's something that you would like to have, but you don't particularly want to ever have to use it. So yeah, that's an innovation in terms of things are not how you want them to be just through market forces. Are you going to have to step in? I mean, it's a little bit related to Martin's point at the start, that people realized that manufacturing was all overseas, and they thought, hang on, we don't really want to be vulnerable like that. We're going to sort of take steps to change that. Not sure I answered the question, but those are my thoughts.
Martin?
I think we certainly need innovation in the area of communication to key stakeholders in funding bodies to understand or accept the evident risks. I mean, we can now reflect on the cost of addressing the current pandemic versus the cost of continuing to fund basic research and applied research into being more proactive in a forward-thinking response. And it's clear that certainly antimicrobial resistance, it's a known problem, it's growing. So how we go about changing the way we message perhaps, or communicating the risks and the outcomes, may be an area where we could see a more proactive response rather than a retrospective one in future.
Yeah, prevention is key also, isn't it? Paul, do you want to reflect on this?
Yeah, so funding for ongoing activities is critical, and we do rely on the generosity of richer countries and foundations and so on, and there are quite a few around, but it would be nice to have a better, more stable funding base. In terms of how to make low-cost tests available and sustainable in many countries, for example, in East Africa, there are already quite sophisticated payment methods which are carried out through mobile phones and through connectivity. So people can buy Coca-Colas and all sorts of other things. We need innovative ways to distribute as well. And one idea that's been put forward is to send out diagnostic tests that are easy to use with the delivery of Coca-Cola, for example. And normally we'd say we'd want those tests to go to a healthcare professional. But if you can make them simple enough, you only have to think about what is the center of communication in some of these communities. It isn't the hospital or a GP surgery. It's people that go to the local shop.
Yeah.
And so if you could make these tests available and workable and connected, then you could have a very novel distribution method and even plug into I forget the names for these methods of payment, plug into the online payment that goes via people's mobile phones. And it's quite amazing just how sophisticated and widespread that is in, for example, Uganda and Kenya, places like that. And we are trying to work with people who can tell us about these methods, these techniques, which are becoming embedded, and help us to find novel ways to get tests to people that need them without absolutely having to go through an established and often fairly sparse healthcare professional network. And I guess what's back to then saying, well, we need the same kind of approach to how you work with the people that are going to use the test. The same kind of approach that was used with pregnancy testing. And I well remember in the mid 80s when we were, and it was a team that invented the technology. It was a remarkably effective team of people that invented it. But the saying was from the skeptics, you will never stop women wanting to have their diagnosis from a GP. They won't want to, most of them won't want to do their own test. And what do we have now? Nobody, almost every woman tests herself. And GP play just a bit part of it, at least until they get into the care through the pregnancy. And it's that kind of revolution where people might say, well, you can't have testing done by tests bought in the local shop or by people that are just not trained. No, we need to use those routes. And if we can make the test electronic and able to transmit the result easily into the mobile phone and able to be collected for epidemiological or other aspects, that's great. We just use the systems that are there and make sure we understand them and know how to use them. And that can take care partly of the money side of it, but also the distribution and collection of results, which are all equally important.
Brilliant. We have a comment in the chat box and say, brilliant point, Professor Paul, for your agreeing with you that simpler, widespread and early detection with a clear plan for escalation of a result will also work a long way to reducing potential stigma of positive diagnosis. So I think we have the last kind of 12 minutes, and there's a question from the audience. Says, in terms of innovation, in terms of engaging with your end users for our vaccines manufacturing and Formologic, so how do you engage with your end users when developing your new products or technology? And I also want to kind of squeeze in a question here about arts, humanity, and social sciences in this area. So whether we should be interdisciplinary and thinking about how to get the wider research community or the wider community to engage with us in on this journey to develop new products and technology. Can I have Marty to have a reflection on this?
Where VMIC is on its journey towards establishing vaccine capacity, we're quite a long way away at the moment from our ultimate end user, which is each one of us. We're doing a little bit at the interface from a technological perspective, just to dovetail on from something Paul was just talking about. We're starting to think about or have conversations with the manufacturers of auto-injection devices for vaccines. So that's one way in which we can be a little bit closer to our ultimate end user, and that is, you know, to ease the administration of vaccines. In developing vaccines, we're always mindful of what's called reactogenicity and pain at the site of injection, and so we can be mindful of how we develop the formulation so that it minimizes pain on injection. So we do that a little bit, but we're not yet at the point where we have big advocacy groups and we're thinking about it, but there are certainly organizations that do that from a vaccine perspective.
I have perhaps a follow-on question for Martin, which relates to this. In my earlier career, I was very much involved with the mucosal immune system, research into it. And given that the virus normally makes its entrance through our nasal and upper respiratory mucosa, is there any realistic potential to do mucosal immune vaccination through things like nasal sprays, so that you actually don't have to inject the source of the antigen, but actually take it through a nasal spray or through something that's eaten and so on, and use mucosal vaccines rather than systemic vaccines?
Yeah,
I don't know the answer. I'd be interested to hear your comment.
Yeah, this isn't an area of expertise for me, as I think similar to you, I'm an engineer rather than an immunologist or a scientist. I'm vaguely aware that there are, you know, second, maybe third generation vaccines being developed that could target IgM responses in the mucosal membranes, and to go after sterilizing immunity there rather than disease suppression. But it's not an area of expertise, so I couldn't comment more than that, other than I think that's where we should be headed.
Yes, thank you.
And also, I don't know whether it's we should discuss this within this group. I was just thinking in today's talk, even for is vaccines actually gonna be helping kind of antimicrobial resistance? If more people or more animals get vaccinated, will we use less antibiotics? I just wonder. Sarah, do you want to?
Vaccinated against COVID, you mean, or
just all infectious diseases?
Absolutely, yes. Vaccination is a reasonably cost-effective way to reduce the number of infections, both bacterial and viral infections. So you're reducing inappropriate antibiotic use for people who would have had a viral infection and inappropriately taken antibiotics, and also avoiding appropriate antibiotic use as well. Absolutely, yeah. Vaccines vaccines and diagnostics very, very important in trying to tackle AMR.
Yeah, thank you. Paul, do you want to have?
Yeah, so in terms of interacting with our end users, we do take that very seriously. And so when we develop a technology, we pay a lot of attention to not having steps within the test that require timing or complex sequences of operations. And so what we try to do is to design into the test itself real simplicity and use, the sort of hands-free almost. But paradoxically, the more you do that, the more you make your test expensive. So if you want a really simple, a really low-cost test, you do need to involve the user to do certain things to make a very simple thing work. So in a way, we have to spend a bit more to make the test simpler to use. And the fact is that the more complex the operation is, the more unreliable your test is. And I think a lot of the reports to do with low efficiency of lateral flow tests is because people weren't very good at taking the sample. So right at the start, you've got a problem that the virus is going to be up the nose or in the throat or maybe in the saliva. And you've got to get that virus into your test. And the interface between the body and the test is a big interface. It's a problem interface. So you have to have extra bits of kit, extra bits of expertise. And maybe not surprisingly, people aren't very good at taking a nasal swab. We're doing without the deep swab. We're now using the outer nasal area, well, not the outer bit, the lower nasal area, and that works better. But I often think of the user who's come into this. I mean, I've lived with lateral flow tests for decades, and I'm very comfortable with them naturally. But when you've got somebody who's never used one before and never had reason to use one, and they're not like a pregnancy test where you just have to put the end of the test into a urine flow, you've got to do something, and you're not really very sure how to do it. You'll be tentative. You'll be wondering if you've got things done right. You might not. We had somebody who had to do a test in one of our studies where you put the swab into a tube, mix it with fluid that's there, and then take the swab out through a squeeze point, and then you've got a sample of extracted mucus and so on, and then you invert it, and it's got a dropper on it, and you have to drop 3 drops into the well. Now, you'd think that was pretty simple, wouldn't you? But would you believe that some people interpret 3 drops as 3 squeezes? So instead of 3 drops, it's 3 squirts, and the test becomes saturated, and it doesn't work. But then why should we be surprised? Because if nobody's ever done anything like that before, why should we be surprised that 3 drops can be interpreted as 3 vigorous squeezes? Very, very different in terms of the amount of fluid that goes on the test. So we have to pay attention to all of these things and try and eliminate all the human error situations that can occur within them. And we do try to do that. And I think we're getting there, but we're not there yet.
Yeah, related to Paul's point on how do you engage with your end users? I think the question should be, when do you engage with your end users? And then the answer is as soon as possible. Yes. Don't say, I have a fantastic product. Why won't anyone use it? Yeah.
And that applies to any diagnostics. And my daughter runs a horse saliva testing service with special swabs that go in the horse's mouth, and there's a handle at one end. And she regularly gets people that send the swab in where they've held the nice soft swabby end and put the handle into the horse's mouth, and then push the swab in back to front in the sample. And these things happen. It's all right if it's a horse's saliva and you're testing it for a tapeworm, you have another go, and it's not an immediate problem. But if it's somebody who's a super spreader who's identified as negative, then you've got problems.
Great. I think we only have 2 minutes left to the end of the session. I must say I enjoyed it very much. I learned a lot from the 3 of you. But I just want to open all the, the floor to the last minute to any of you to want to make final remarks about this session. And maybe I just want to remind the audience the last session is tomorrow. And please join us when you can, if you can. Yeah. So any of you want? Yes, Sarah.
I can start if you want, just for final remarks. So regarding the session being about knowledge exchange, just to point out that knowledge exchange includes people sharing negative results. Well, depending on what you mean by negative results, results which weren't what you had hoped for, let's say. And to mention one particular initiative, that's Spark, shared platform for antibiotic research knowledge. So the idea is that when R&D projects close, which they will, the high risk projects, you know, the science just doesn't work out, that data would get uploaded for other people to share. And I like this this expression: don't repeat old mistakes, always make new ones. So, you know, it's just a waste of money and waste of everyone's time and effort trying something someone else tried 2 years ago, 5 years ago, and it failed for XYZ reason, and they didn't tell anyone about that. That's my overall comment.
That's a wonderful thought to take with us.
Good. I think we're just at 15:30. Maybe we shall end here. Thank you again, our panelists, for the wonderful session. Thank you.
Thanks for the opportunity.
Yeah, very enjoyable. Thank you.
Such pleasure to hear from you all. So